partner och sätt att jag tycker inte.
Everyone is heartily welcome to this session, and today Moiz Bakhiet will defend his thesis on a subject, and the title of the thesis is Immunopathogenesis of Experimental African Trypanosomiasis. This is the last stage, or the last event, to get the Swedish medicine doctorsexamen. And as a opponent today, we are happy to welcome Marita Troje-Blomberg from Stockholm University as opponent, who will critically scrutinize this thesis, I hope. And there is a committee who will also judge the defense and the thesis, consisting of Professor Åke Seger, Geriatric Department, and Docent Åke Sidén from Department of Neurology, and then Lars Rombo from Department of Infectious Diseases, and Professor Mats Wahlgren, recently.
I feel really very sorry for those errors that I couldn't detect before printing the thesis. And I would like to distribute them. I will pass them.
The trypanosomes was first discovered around 1880, and the first species of trypanosome discovered was Trypanosoma advanced, which was found in the blood of camels. Then later on, the Trypanosoma brucei was isolated and shown to cause sleeping sickness in Nagana in animals. The Trypanosoma rhodesiense and gambiense in humans was not discovered until 1803, while one has 1903, while one had known the disease for at least 100 years. Most of parasites have been. If we look here for the life cycle of trypanosomes, you will see that when an infected tsetse fly bites a mammal, either a cattle or a human being, you will inject a long, flagelated, slender form, which will multiply and divide, and then end up in a short, stumpy form. This short, stumpy form will be taken up by the tsetse fly, and in the fly you will have a morphological and antigenic different stages of the parasite. The parasite will go into the midgut and will divide repeatedly and form a so-called procyclic form. This procyclic form will continue to divide and become an epimastigote, which will go up to the salivary gland and then form a so-called metacyclic form that will be infected for the human. If one look on the surface of this characteristic parasite, you will see that the major component is a variable glycoprotein, which is called VSG, which is a huge molecule, and it's about 10% of the protein content of the whole molecules. These VSG antigens has the characteristic, or the parasite has the characteristic to be able to change the composition of these antigens and thereby sort of evading the immune response. These VSD genes are present in the blood-formed stages and in all different stages in the mammal host, while it's absent until in the tsetse fly until the parasite enters the stage that's infected for the human being. It's known, or it has been shown, that these VSD genes can be attacked by the immune response by antibodies. And by antibodies you could eliminate almost 99.9% of the parasite, but still the one parasite that will survive will have another set of, be capable of producing another set of VSD genes and thereby not accessible for the antibody-mediated immune responses. And since it's divided, you will have a new peak of parasites. So what other ways could the parasite then escape the immune system? We have already discussed this exchange of immunodominant surface antigen on the parasite. One could also have, or the parasite could generate post-antigenic mimicry, which then means that the parasite sort of either absorb or produce molecule so that the immune system cannot distinguish between cell and non-cell. The parasite will or can induce polyclonal activation of the immune system and thereby having activation of many different cells, which then will generate the immune system to produce autoantibodies and antibodies against self-components, and thereby the whole immune will block the effect of IL-4 and thereby diminish the antibody production, which could be possibly what is happening in this disease. So this is my brief introductions. I leave this to you guys.
We are looking on the topic. And as Marita said, due to the living conditions of parasites that they need humidity and hot temperature around 40 degrees centigrade, the parasites or the vector is limiting itself into areas extended between the latitudes 14 to the north and 29 to the south, and limited by eastern and western coast of the African continent. And in this area, around 38 countries and around 55 million people are at risk of getting infection. And every year, around 30,000 are reported. And in the WHO, they are telling this is maybe very much less than what is true, because there is no regular studies and follow-up in those areas. And unfortunately, nowadays in Zaire, there is the severe outbreak of trypanosomiasis, and nobody is taking care about this severe outbreak, and almost in the villages around 20% of the people were infected. This is the taxonomy of the parasite, which I will not go into details, but as Marita said, there are two main subspecies are causing disease in human. Trypanosoma gambiense in West Africa and Trypanosoma rhodesiense in East Africa. And Trypanosoma brucei is causing infection or trypanosomiasis in animals. And this is frequently used for experimental systems. The parasite is an elongated parasite. It is around 10 to 40 micron, and it has a nucleus and a flagellum and a kinetoplast. And usually the kinetoplast is a very specific from here and onwards. This TFTF binds to the CD8 T cells and triggers the CD8 T cells to produce interferon gamma. It's known that interferon gamma is a cytokine, usually with protective effects. But instead, the parasite uses this protective factor as a cross-promoting molecule. And then, instead of being killed by this one, it proliferates or increases in numbers and causes more disease. The aim of the study is to characterize this factor, and then to study the selective role of the CD8 T cell or the CD8 molecule as a target for the factor, and the way the factor stimulates the CD8 T cells to produce interferon gamma. That is to say the intracellular signaling. And then to study this interferon gamma, the production of interferon gamma and its role as a cross-promoting molecule. This study started by an early observation that the parasites do not penetrate the blood-brain barrier, but they are restricted into areas outside the blood-brain barrier, like the circumventricular organs. For example, the neuronal lobe of the pituitary gland, the vascular organ of the median eminence, the subcommissural organs, and pineal gland, etc. So those areas at the same time shows infiltration of parasites and inflammatory cells, and mainly the cells which were dominating here are the CD8 T cells among other cells. And inside the brain, there was induction of MHC class 1 in an areas do not lack the blood-brain barrier. And this is, for example, in the paraventricular nuclei, here is the third ventricle. And interestingly, those areas are projecting their axonal terminals from inside the brain. So, since CD8 were dominating among other T cells, we plan to study the role of these CD8 T cells. And to study this role, we decided to deplete the cells. And by depleting the CD8 T cells by injecting monoclonal antibodies against CD8, then this MHC induction in the paraventricular nuclei was abrogated. And since interferon gamma is the main inducer of MHC class 1, and CD8 T cells are one of the main inducers of interferon gamma, we studied the interferon gamma production during infection. And very early during infection, only 12 hours, there was a dramatic induction of interferon gamma, just 12 hours after infection, and it kept also at a low level compared to the controls. Whereas CD8 depleted cells, animals, did not show any increase of interferon gamma. They were as control. Also, the depletion of CD8 T cells lead to decreased number of parasitemia and prolonged survival of the animals. So at this stage, we could conclude that depletion of CD8 T cells abrogated MHC class 1 induction in the brain and interferon gamma secretion or production, and decreased parasitemia and prolonged survival of the animals. So CD8 are crucially important during the disease. Then we have studied this mechanism in vitro, where we co-cultivate parasites with mononuclear cells from non-infected animals. And by doing this, the parasites were able to stimulate the cells for production of interferon gamma in vitro. And also we used a method that could detect if there is any diffusible factor passing soluble CD8 also block the effect of the TLTF by competing with the factor. And here on human cells and here on rat cells. Then to directly study if there is a binding or a direct binding between the TLTF and the CD8 on the surface of the T cell, we used 3 different methods. The first one is in ELISA, where we coated the plate with. The second method we used is immunohistochemistry, and those are the same cells, air dried in slide, and then we used MO1 and stained by immunohistochemistry, and then we could see more than 70% of the cells stained by the anti. And I have forgotten to tell, this done by the way that we add the factor to the surface of the cell and let the factor interact with the surface of the cell, and then use the monoclonal antibody, the MO3, that will label the factor if it already bound to the surface of the T cell. So we use this MO3 both in ELISA and in cytohybridization, and also in flow cytometry that I don't think I will show. Here are the CD4 cells which do not show any stain. Also, we use the genetically manipulated mice. That is, those mice lack the CD8 genes and they are lacking the CD8 T cells. By infecting those mice could survive longer than ordinary infected mice have both CD8 and CD4, and showed continuously low parasite FYN. And usually, the T cell receptor with the co-receptor, the CD3, are usually transmitting signals through protein kinases families, that is protein kinase C and A mainly. And this is the classical pathway for the microgenes to stimulate the cells. But this study we have done is to show whether the factor is using the classical signal by the TCR, the T cell receptor, and the co-receptor CD3 and stimulate and activate the protein kinases, or it uses another signaling mechanism through the CD8 and the tyrosine protein kinases. Because the signaling is through 2 specific pathways, either protein kinases C and A, and that is the classical, or through tyrosine protein kinase, and this is used by anti-immunoglobulins and IF2-like growth factors. So to do this, we use blockers, drugs, that block protein kinase C, that is H7, and protein kinase A, that is H8. And this is a classical pathway. And we use interferon gamma production and proliferation. And here, those blockers did not block the effect of the TLTF, either on interferon gamma production or in proliferation. Whereas they did block the effect of the microgene, the PHA, the lectin here. But the other signaling pathway, we used genistein. And then genistein, that is for tyrosine protein kinases, blocked the effect of the factor. Whereas also here, the PHA was blocked by genistein. And this could be bypassed for all esters, that is bypassing the T cell receptor. So this is not due to non-specific toxicity. From this, we conclude mice to study the role of the gamma delta T cells. And also nude mice were able to produce interferon gamma. And by using in situ hybridization technique, we looked for in vitro by cultivating cells from those different mouse strains with the factor to look for TGF-beta and interferon gamma. And also all mouse strains showed reduction of both TGF-beta and interferon gamma. But IL-4 was only produced by in vitro by the C57 black interferon gamma reduction later on.
then if we go to paper number 2. I would like to clarify how we prepare the antigen fractionation that were used for gel filtration. Yeah, because it's for me very confusing.
We have some experts here helping out. So this is by using gems that can separate molecular weights from 10,000 to 200. Then just disrupt the parasites by ultrasonification, pass through gel and connect to spectrophotometer and then collect 120 fractions automatically. And then this is, because this is separation according to size and molecular weight. So we collected those 120, and then I took those 120 and examined every fraction, or it's a B fraction. But if there is a good method to screen, then you could know which factor, which fraction is of importance. And then there was really very high fraction with a very high T.
That's what I'm confused about, because when you extract the thing you put on this gel, I mean, I do understand the size fractionation and the idea of testing the different fractions. But it said here that you take your cheek brucei and you sonicate them on ice for 1 minute, and then you pellet the material by ultracentrifugation at 3 hours for 105,000 G, which is going to separate your material into soluble and insoluble fraction. But then, as it said here, it's the insoluble fraction you work with. Is that really the case?
They are soluble, which we got from the.
But as it said in your material methods on page 2448, under preparation and fractionation of T brucei, as it said here, it's that you take the pellet.
Yeah, from after centrifuge.
You do not take the supernatant.
No, the pellet.
So this profile that you will get the high molecular weight must be complexes of your factor composed to a membrane fractions and things like that. Because usually when you make a fractionation, you take the soluble fraction and fractionate. Because otherwise you would have membranes.
Yeah, but we tested this for ability to use it at the end. So we didn't just throw the supernet, we kept it and we tested it and we were sure if there is any factor in that producing the field.
So there was nothing in the supernatant? It's only this membrane?
No, it's not nothing, but you cannot dilute out. It's, you could find some activities in the supernatant. If you put it as it is, but if you dilute it out, then you don't see any.
Is it because it's too little or?
Yeah, because maybe there's no, the factor is not there, membrane bound or something like this.
That's interesting. Because later on, I mean, the purified material we get, which is 45,000, that must be a soluble product. And this soluble product is that active?
Yeah, but I don't know how this, because we don't know where is this factor. Is it in the staircase, whether it is in this flagella, from where it comes, we know nothing about the factor coming from the parasite. And it could be a membrane factor and could be secreted by some mechanism during in living parasites. But later on, when doing fractionation, we got the activity on this part of the as a pellet after centrifugation.
Because in the first fractionation, it's something molecular weight 185,000.
Yeah, but we got other, this content could be multimer and aggregates, and then we found some small activities in other fractions.
So don't you think that this the other fractionation that will tell you that the factor somehow is associated with membranes or something like that, your way to solubilize this from the parasite was not good enough, since you could not detect it in the supernatant. Because you have but much lower.
Yeah.
But that this high molecular preparation contain other things too than the factor.
Yeah.
So if we look on figure 1 in this paper, where you have tested the number of interferon-producing spots in response to different numbers and fractions of the parasite in non-depleted and CD8-depleted animals, both in the lymph node and in the spleen. The one result that you say there or tell in the result part is that in the spleen cells, large number of parasites will depress the immune response or the induction of gamma interferon. But since the parasites are dividing, so how many numbers do you actually have in these fractions? And how do this number then correspond to this 10 up to the 4th frozen trypanosomes that we have used as another source of antigen? And this frozen antigen, was that titrated or?
In some way, I mean, we calculated the number of parasites in the beginning before they die, and then we distribute them into different layers. And we know from this how many parasites are there. Then it's approximate calculations, it's not a data. And then here for living, I mean, we have 21 down parasites that could give stimulation for T cells. But I thought easy to tell whether it is 1 or 10. So we didn't show the 1 because this 10 could not be 10, it could be 5, could be 15. And this is by limited dilution, putting the number of parasites. But we think that those are variable parasites that they could really induce better than frozen, because when you freeze, you could use some of the activities.
Or maybe it's so that, as in Leishmania, that live and denatured parasite antigens do induce different types of immune responses. Because what you have to say is that with the frozen fraction, you would never get as many spots as with the, even if you increased or decreased the number of parasites that you use for the frozen material. You could never get the same level for
living parasites.
or as compared to what you use.
Yeah, living parasites are giving better stimulation, but also this depends on the time when you use those living parasites. You use them as early or later.
So if we look on figure 3, where you also have used these different numbers of trypanosomes, have you ever tried these antigens or the different numbers of trypanosomes for cytotoxicity or toxicity? I mean, if you take a pHA response and add the trypanosomes, what would happen if you add trypanosomes?
To PHA.
Yes.
I don't remember that I did this experiment, but I mean, I used PHA just as a control, as you see here. Yes, but I mean, I didn't. But another thing that we do, because here you see low proliferation by the parasite, but when we used beta responder cells from CD4 deleted mice that has only CD8 population, as in the paper 5, then we saw much better proliferation, and that could be due to the increased number of the responder cells, the CD8 T cells, because in this MLC, the CD8 could be 20%, but in that one could be 90% or something. Then we got better proliferation. But what we did is we tried to block the interferon gamma if it is anti-proliferative, has an anti-proliferative response by adding anti-interferon gamma antibody. So then we could a little increase the proliferation by the fragment. So in the beginning we thought that this low induction of proliferation could be due to downregulation or inhibition of proliferation by interferon gamma that induced by the fragment.
The reason why I'm asking this about toxicity is, I mean, in this figure, the proliferative response is low, but it's known that trypanosome infection would affect IL-2 receptor expression and also IL-2 release. So if you would have added the trypanosomes to the PHA, you could somehow tell if that was toxic or other things are happening. You have not added IL-2 to your...
No, but I thought this is due to proliferation because of the ability of the factor, not due to toxicity, because the factor was able to induce the same cells to interferon gamma. So I thought, I mean, I didn't think about toxicity and that since the cells were not dying in terms of interferon gamma. This is why I do think that type of
it's known from other systems that gamma interferon proliferation is not associated. So what do you think, or do you think that what you see in these two assays are activation of different subsets of cells, or how would you interpret the data?
Here in the beginning I thought this is due to ability of interferon gamma to inhibit the proliferation.
Of the same cell or another type of cell?
Of the same cells.
Why wouldn't that be the case for PHA? I mean, I would have predicted that by adding anti-gamma interferon to the PHA culture, you would have an enhancement of this. But why wouldn't gamma interferon inhibit at least something?
I mean, sometimes the factor, if it is really active factor, stimulate much higher than pH. I mean, we saw the factor, if it is a good preparation, then it is more than 3 times than the pH inducing interferon gamma could be high induction.
Then on figure 8, it's not so nice. Where you actually have made a peroxidase immunostaining showing that the parasite seemed to take up gamma interferon using a rat anti-rat monoclonal antibody. How could you say that it's not secretion from this?
From the parasite, no secretion.
Yes.
I did one experiment. You put the parasite like cells, but I was doing a lot of control experiments to see if really they are secreting or not. And then also we extend them for hybridization, put them beside the cells for other purposes. We wanted to see parasite beside the cell. But we saw the cell hybridized, but not the parasite.
So you think it's actual uptake? The parasites will contain or express epidermal growth factor receptors. Does gamma interferon in any way interfere with those receptors? Or any other receptors? Do the parasite carry any receptors of anything?
Yes, they are carrying other receptors for other transferrin.
Will they be enhanced or why is gamma interferon promoting growth?
But other factors are promoting the growth of the parasites, this epidemic growth factor, other transferrin, cysteine recently, promoting growth.
But what I'm at is that since the parasite actually is taking up the gamma interferon, so is the growth promoting effect of gamma interferon, or is gamma interferon interfering with enhanced expression of receptors, or
we think that it is a receptor for the interferon gamma. That's what we are looking for now. To see if there is any receptor. But how the parasite take up the interferon gamma?
and what is the actual effect? What do you think?
We did one pilot study very recently, because we didn't work at the level of the parasite itself. But the only thing that we did, we could inhibit the effect of interferon gamma by growth stimulation by inhibiting signaling pathway, because we saw a report that those parasites showed some protein kinase, tyrosine protein kinase activity. And then we tried to block the effect. I mean, that was the first pilot study on the parasite itself. But really we didn't know what, or we didn't work actually at the parasite level in a way that I can explain. But I think that we really do if we have future plans. We have to see both arms of
We need to expand the group and work in many different directions. So if we go to paper number 3. Figure number 1.
commonly used. But the purpose here was just to see that not due to toxicity of the MO1. So this is why we used this PHA. We didn't go into others to show that MO1 is not toxic, that it could be bypassed by PHA.
When you prepared your monoclonal antibodies, then there is you also used some trypanosome antigen fraction, which is also
I coated the plate with MO1 with different concentrations and to see and as a control the DB1 of course. But then MO1 did not detect any single spot. That is, in those ways, we try to do some independent studies. And since also MO1 did not inhibit this induction, as PB1 of class 2. Then we thought it's not cross-reactive.
And then there is in this paper for
many different controls are doing this. For example, by adding SDS to pH, or I don't remember exactly what, but we did so many controls to try to get an evidence that it's glycoprotein, or to characterize more characterize the factor. But of course, this is this we think it is from this is glycoprotein. Still, we have to further characterize this factor.
so then if we come to the figures 11 and 12. And in this experiment, you have taken animals infected with trypanosomes and distributed your neutralizing monoclonal at different time intervals. And you have also compared these. The readout here is the number of trypanosomes, and you have looked for the survival of the actual survival of them in the same animals. If I look in the non-treated animals, which is here, you have fluctuation. So is these fluctuation antibody neutralizing of trypanosomes, or what is?
Yeah, this is known. You get this kind of up and down in urine, and this is mainly due to antibodies, anti-BSDs antibodies. So it is this. So you get first parasitemia, and then you get depression, and it is due to antibodies. That is the main host defense. And then later on, when you have immunosuppression, then it must be constantly high. But it's going like this. 2-3 waves of parasitemia coming, and they are depressed due to antibodies, and when the parasites switch.
For the actual binding of your factor to CD8, the CD8 molecule. In figure 2, I have one comment. In this part of the figure where you have tested the effect of your factor and CON-A on mononuclear cells, and as we have had interferon gamma, you'll find that both your factor and CON-A induce a large amount, and you show very nicely on your summary that the conventional way for a T cell to be triggered, or a T cell, or a cell to be triggered, is through an antigen and CD8 or CD4 positive molecule, and then you will have a calcium-dependent activation of serine or threonine protein kinase family that will be involved in the signal transduction. Or you could have the calcium-dependent protein kinase A, the PKA system. This is also holds true for PHA activation, whereas if you look for I2 or anti-ID, these are usually PKC and PKA independent, but instead use protein tyrosine kinase as signal transduction. And you have shown then that your factor induces this system. But in your inhibition experiments, you do not get a complete inhibition. Why?
Could be another pathway. I mean, especially with proliferation.
Could it be that you have
we thought about this, and could be another signaling involved. I mean, I really don't know why we didn't get back down to complete inhibition. And I think we mentioned that in the discussion.
Do you know of other systems? Because this is strange. Because you can block your binding with anti-CD8. So do you know of other systems where you could have actual binding to one receptor and then go to another signal transduction situation?
Other than those two specific.
Yes. But actually you refer to a paper by Rurke, and interestingly enough, that the same signal transduction pathway that you are seeing, they have seen used in recognition of alloantigens in the mouse system. Have you ever tried to block the binding with anti-class 1 of your molecule with anti-class 1 monoclonals?
To block the binding?
Yes.
Between the factor
and the CD8. But when you look for your factor binding on the CD8 positive cells, which you could block them with anti-CD8, did you try anti-class 1? I think that would be interesting, because this is what they find with this allo recognition. This could be blocked by anti-CD8 molecule or antibodies. And the signal transduction pathway.
Cross through.
Yes. If we then go to paper 6, where you change completely to mice. But not only that, because in all your earlier papers you used male rats, or so mice that with females. Are there any sex differences? Or
No.
So why did you change?
In the literature, because we wanted to do the same, to use the same pattern of disease. So in this paper, we tried to do an experiment done by another group and repeat and see the role of the bacteria. They were using females, so this is why we use the same.
And in this paper, you're going to look for animals. Yes. I would never, and you should look for subclass distribution, which would be very informative. The only thing I wonder then also from this paper, since you would want to look for I4, do you think that is the best cytokine for looking on the T2 type of cells? I mean, I4 is a switch factor. It's not really a B-cell regulator factor.
Yeah, we also plan to do profile for cytokines. We will use in cytohybridization now we have other crops. And that's also, we will do this cytokine profile. Of course, one should look for IL-5, IL-6, on pH2, and some people have looked on DNA alpha. So I mean the cytokines which are important in antibodies production from pH2. Now we are looking for IL-4 because we found this invention and this difference, so we would like to extend the study. But of course we should look for others, IL-5, IL-6, and maybe later on IL-10 and IL-13.
So then, finally, I will finish by telling me from your, asking you to tell me from your model. I have heard that you have been out in the newspaper telling that it could be possible a vaccine, your molecule. And then I would like to ask, in trying then to have not only the molecule but this information as a tool to diminish or eradicate the disease, how, on what level would you go and how would you do it? Where are you going from now, so to speak?
I think this model could be a work for 10 years. Only this model. First, this factor. I will concentrate on your question on the vaccine. So I think when one considers to have a vaccine, then should identify the molecule or the factor which we will use as a vaccine. And here I think on our hand we have one factor, but we need another factor. So I will start with it. For example, this, I will come to.
I just asked something about this factor. You have the factor and you have it purified and you could generate monoclonal antibodies against this factor. How is it doing, which were made in mice, the monoclonals? How is it in the natural situation? Because this molecule should be produced all the time. Would you
yeah, we did experiments using other parasites, not living, frozen parasites. And we used antibodies against the factor. And all parasites, all antibodies goes to different parasite strains like Gambiense, Rhodesiense, but only one which the parasite, the antibody did not block its ability to induce interferon gamma was advanced in the coming. And now we are going to extend this work on this parasite. Maybe it has different.
But it's not in general.
This was very good for us. That this factor is a candidate for vaccine. In addition to treatment at different time points, at least it is invariable. And I think this is the only factor reported to affect the survival of the animals by blocking the factor.
But you will not, during natural conditions, get natural antibodies.
It should be, and that is one of the things we would like, because the diagnosis is difficult, and we would like to use an ELISA system to detect this factor if it is secreted early, to try to diagnose by this factor if it is shared between all different parasite strains. So I think this factor, because of those facts, it's a very good candidate for a vaccine since it is invariable. And then we need, of course, to sequence this factor and have it cloned. And another molecule, I mean, this block, the part of this model, that the parasite secretes this factor and goes to the CD8 and trigger interferon gamma. But of course, when the parasites are infecting human beings they will be already interferon gamma from other sources. And then if the parasite will be able to pick up those. And the second thing will be the receptor. I mean to block the two pathways, to block this triggering part and to block the uptake if it's taken by the parasite. And the idea of this is that to reduce the number of parasites so that the humoral mechanism could find its way and kill the parasites before they switch to another or to increase in number. So the first thing in doing a vaccine is to have this factor clone, sequence and clone. And then one generally think about the vaccine should think about to identify the protective immunity, whether this will protect, as I said, against the parasite itself or against the disease, or what kind of protection will come out of this. And then it's important also, I mean should be very extensive work to see if this immunity will not, if this vaccination will not harm the host. I mean one should test because it's known from other systems like in viruses, in lymphocyte, choriomeningitis virus. Those are usually not harming in the animals. They are sitting like the carry. But if you immunize them, you will get opposite effect. You will harm the host. And this is the case also for food poisoning with staphylococcal antitoxin. And sometimes you get viral enhancement, like in dengue fever. You just block the virus by immunity. So one should really know this kind of immunity will give protection. And then another strategy should be that to identify the immunogene in a way that whether it will be T-cell dependent, and because T-cell are the fundamental cells that house in memory, so you will get good memory or an elastic response, and you will get IgG response with long lifespan, and also they are living longer, or you will get B-cell response with IgM with antibody, or you will get T and B cells, as I have seen in one of your papers. And then the chemical structure. If you are going to use it, is it a protein, glycoprotein, or now people using anti-idiotypic vaccines? They are the internal image of the antigen you can use. And then one go on try to do how to inoculate when, and using adjuvant or using allo. I mean, different things one could think. And I think it would be a very long project. But the crucial thing now is this: to pick this and to sequence and clone this factor. This is very important, and to find the receptor here and also sequence and clone the receptor.
Do you think the parasite produces the factor just activated T cells itself, or do you think that it somehow it's essential?
I think they are. This is one thing, but also they could use this factor to reduce this polyclonal activation. For example, in cruises, a lot of discussion. If you could prevent this or reduce this polyclonal activation, then you could give better control. So the factor could lead to this polyclonal activation.
So I'm finished, and I would like to thank you, Moiz. I learned a lot during reading your thesis, and I really enjoyed the discussion with you. I congratulate you to a wonderful and exciting work. And
Well, unfortunately, we are not ready yet, so perhaps you should wait. But now there is time for more questions, and the custom is to start with the community. Are there any questions? Any comments?
maybe I could start and ask about the parasite then. And you work with Trypanosoma brucei, and I wanted to know how good is this as a model system for the human disease. How you sketch briefly on the human pathology, but go deeply into the experimental pathology. And how is it actually so that in the human case you also have a restriction to areas outside the brain-blood barrier as you have in the experimental model system?
Yeah, I think so. It's the same case. I mean, and the pathology in the human and animal system is not really big different. I try to see so many papers telling about pathology in animal, and that is very well studied, more than human. And
that's my question actually, because you describe the pathology in the human very different from the mouse. In the human, you see the morula cells, the plasma cells, the IgM, or the sort of dominant, and maybe some lymphocytes infecting. But the main.
Yeah, because this is very well studied, and many people work with this. And I mean, one of the things now, my supervisor, Professor Christensen, started to look for the EEG pattern on animals, because he had this feeling also. Last time he told me he's very happy to find the same pattern of EEG changes in animals and rats like changes in humans. So he told me he's happy that this model is good for
So then, do you think it is a good model? Do you see the same cell infiltrating the human brain? You see the CD8 positive C cell infiltrating the areas?
in human, I don't think there is. I mean, I haven't seen report with CD8 infiltrated.
Because this is a very important point, I think, because many experimental systems the parasite used is different from the one used in which causes disease in humans, and then the model system actually that is sometimes irrelevant. I'm not saying this here, I'm just saying that it's an important point to discuss, the relevance of the experimental system for the disease, and also why do the rats or the mice die, and why do the humans die? Do you know that? Could you talk about that?
Why they die?
Why they die?
I think the most common cause is the secondary infection, because the immunosuppression, and the most common cause of this is they die due to secondary infection.
So that's the same for them. I wanted you to compare the mouse and the human.
Yeah, of course, this is very important, and also we plan to do studies on the human. We have some contacts with institutes in Africa because the ultimate goal is the human being for us, and then we use this model as we can study this interaction here in the lab. But then, of course, if this model is not applied to the human being, it's no meaning.
Another thing I was wondering about what happened to the NK cells in your studies. I mean, a major producer of gamma interferon is NK cells, aren't they? And they are approximately 10% of the CD8 lymphocytes is like 10-15%, and NK cells are triggered, for example, by IL-13 to gamma interferon production. Could it be that there are other factors involved in the steps until gamma interferon is released and used by the parasite? Could there be other factors? Could it be that the factor triggers CD8 or actually triggers CD8, maybe IL-13 to trigger other cells or NK cells to gamma interferon?
Yeah, could be. I think we did not study NK cells, but we think they are one of the cells that are triggered at least later during infection. Besides the gamma delta cells and even CD4 cells. So we consider the NK cells could have an importance, especially on those CD8 deleted mice. When we get an increase later on, then there are no CD8 T cells on these mice. That means other cells are important. And one of those candidates are the NK cells plus gamma delta.
I was thinking about the last day, but the nude mice, I mean, they
Yeah,
lack many cells, but they have NK cells.
NK cells.
They survive for 17 days, and there's, would you think that if they lacked NK cells as well, would they survive much shorter than, or do you think?
I mean, if they lack NK cells and gamma delta, they could get support also, because there is another thing that was not included in the video, that is the interferon gamma-like material in the neuronal ganglions and the sensory ganglions. That is also support growth of parasites. So, I mean, it's not a simple thing, so it's many other things are there.
Those fascinating studies, of course, new fascinating questions.
Yeah.
So now I have another fascinating. I was wondering about something. In the paper 4, in figure 13 is actually the last figure. You have CD8 negative mice that lack, but they have a knockout to the CD8, so they don't have a CD8 cells. But still, the mice always die, no?
Why they die?
Yeah, why?
I think because this state of immunosuppression already and pathology there. I mean, this after you neutralize at the later stage, then you cannot restore this immunosuppression back to normal. I mean, the disease is already so far advanced at the time we neutralize with anti-interferon gamma. And therefore, at that time already immunosuppression, polyclonal activation, and a lot of pathology are there. So the animals will not be able, by just neutralizing the interferon gamma, to come back to the immunocompetent level.
Couldn't it be another possibility also that the parasite varies, that actually throws off this factor and uses another mechanism of growth, and then it kills the host?
Yeah, could be.
You haven't studied that, or do you have any indications?
No, we do not.
Another thing this is a very speculative question. There were just some reports in Science that there are gamma interferon gene knockouts now, and also gamma interferon receptor lacking mice. What will happen to the disease in gamma interferon lacking knockout mouse? What are the speculations?
Of course, they will be very interesting to get those mice. But the other thing is this interferon gamma-like material, because the parasite could get support from other places other than interferon gamma from T cells. And we have heard from Professor Bixel it's not easy to get those mice, some company. So we are very willing to have those mice.
But what do you think would happen?
It's difficult to tell, but I mean, those mice, if they don't die from parasite, from trypanosomes, they will die from other things. It will be difficult to tell because they need interferon gamma for protection from other diseases. And if the parasite find a way to live by using other sources for interferon gamma-like or interferon, so then they will kill the animals. It will be very interesting to look at those animals and infect them and see in sterile conditions.
I mean, the gamma receptor deficient mice will be overfed and fat. Okay.
It was mentioned that luck is very important in science, right? You said. Another important factor is fantasy, right?
Yeah.
But I was just wondering, because regarding the aims of the study, because you said the first aim was to purify and characterize the trypanosome-derived factor which interacts with CD8 positive T cells and so on. Now, when you started this study, you didn't know about the factor.
No.
And less so, you knew about possible interaction with CD8 positive cells.
Mm.
That's why I wonder, how could that be an aim of the study? That was something that you did.
Yeah. No, but when writing the thesis, I put it many aims. Started from zero. Study the role of.
But when did you start with the aims? That's when you sum up the.
You don't know. No, because I tried to write the thesis like a story from literature, and then I give a background, and then I try to put big main aims that going together. Aims, result and discussion, conclusion. So you don't lose the way between them. But when you read the first one, if you go back a little to literature or introduction, you will try to pick this. So I put it as a compromise, whether I start from beginning and start results from different area and discussion from different area and conclusion, and or to put them in a sequence that you will never lose. If you read the aim, the first aim, and you open the first part and result and discussion and conclusion, you will find the same. So I try to put them in a clear way. I mean, if I thought if I start from beginning, I will make some confusion. Maybe I am wrong, but but this is why I try to put them in a circle. We put this model here as aim, result, discussion, conclusion.
Let me interpret this in this way, that the aims were more or less constructed or a result of your results. Do you agree?
Yeah. The aims are not only that, discussion and conclusion, everything.
Okay. Now, let me put it this way. What initiated this stuff? Why did you start?
Because of
what were your aims at the beginning?
Pardon?
What were your aims at the beginning?
Because the early observation that only CD8, among other T cells, were expressed in the brain. So that was to study the role of this CD8 by depleting them. So that is the first. We didn't know, and that was very good because I thought by depleting CD8 the animals will die. And by infecting the animals, they live longer. I said, this is wrong. Repeat again. Then again, they survive longer. So I was trying to find the opposite. And then it turns out this is true. And then the work continued.
And then you got the idea with the factor. And
yeah. And then we did this double chamber system to study this interaction. We just incubate the cells and the parasites in vitro. And then there was a very interesting thing. Cells up induced for interferon gamma production. And the trichinosomes down increased in number. Although they are not in contact with each other. So this was the idea there. Then there is something usable in a bidirectional way between the cells. And then we started to think first to characterize this. The best way is to have a monoclonal. And then comes the luck. Of course, with a lot of hard work. But the luck also helped to get the antibody that is really specific for the factor.
Okay, so finally, did you suspect the existence of a factor when you started the study? Did it come as a surprise to you, the co-workers?
I mean, when I thought about it and I did the experiment, then I was convinced there is something. And then I tried to fish this out.
I'm a bit confused of your way of handling the expressions, growth, inhibition and survival, and what you actually mean by this expression when it comes to the parasites. To start with, in your system, how rapid do these parasites multiply? How rapid do they divide?
In vitro,
In vitro.
in 2 to 4 hours, they increase in number.
They increase in number, yes, but how rapid do they divide? Is there a division time by 2 hours?
I mean, this increase of number, if it is due to division or due to the cell cycle itself, then they increase earlier. And this is also was a little problematic in the beginning for us because in Rome papers it was they divide after 6 hours, I think. But then we tried very hard to see if this is true or not. And then we counted, we tried the psi median incorporation, and we really find them increase in number earlier than 6 hours as reported by now.
So you have a division time. Each parasite becomes 2 parasites in 2 hours time.
I think so.
How do you then establish that they stop multiplying?
In vitro.
In
That is difficult because they disintegrate after some hours. So it's I mean, we didn't have the plan to have long culture time. We wanted to see just these interactions. This is a growth factor. They increase in number with interferon gamma, but not without interferon gamma or not.
So your definition of inhibition is really that they disintegrate, they die.
No, inhibition, that means if you add a monoclonal antibody, then it blocks this increase in the 2 hours.
Yeah, but you said it was different to establish. So how do you establish that they are inhibited?
Here is the parasite number. And here is time. It's 1 hour, 2, 3, 4, etc. If you apply 1 million here, per ml, and then in 2 hours, by adding interferon gamma, the parasite go like this. Okay? And then I was not looking then because they disintegrate and die there. And I was counting only living parasites here, not dead parasites. Okay. And then if you don't add interferon, you just put medium without any fetal carb serum, without anything. What happens like this? They die. If you put fetal carb serum, then they do like this, they die. And if you put cells to this medium without recombinant interferon gamma, the same could happen with cells. Okay? But if you add a monoclonal antibody against interferon gamma, then this will happen like this. Or some way.
Which means
That means I got inhibition. That's what I mean by inhibition. So this at the maximum number that was blocked, so the parasites did not increase in number. This is what I mean.
and if you didn't apply anything to the parasites, they would have continued to grow forever. Or
If
you didn't add anything to them,
no, they die after.
they die all the same.
Yes. After this.
Is that why in page 48 there is no virtual difference between what happens after 4 and 16 hours? You see approximately the same results. So they stop multiplying all by themselves.
Yeah.
Well, I have a question related to virulence of the parasite. If you make a second passage through a rat, then the virulence is increased.
Yeah.
What is the probable or possible mechanism behind that? Is that via interferon gamma activation, so to say, of the parasites, or?
No, I don't think so. I think the parasite get adapted in the host, and then they become more monomorphic. And monomorphic is defined that those parasites are not producing stumpiform. And this stumpiform is non-infected and is not dividing. So the parasites you always making cylinder form. And that means they are invading and multiplying all the time and become more potentially virulent and can cause more disease. So this is related to the parasite itself and adaptation in the host.
No, that it is kept in a more suitable milieu, so to say. You can.
So they get adapted, so they are dividing and know how to evade the immune mechanism.
Okay. May I ask you have a monoclonal antibody now for isolating TLTF. Have you made any preliminary immunization experiments with material isolated by MO1?
No, we didn't do that. We postponed this to have the sequence and try to use peptide or part of the peptide and do a lot of series of experiments. But I mean, one thing to do just in infect the animals is a TLT and see what will happen.
How long before you will have the sequence?
We hope we could have it already now.
Ah,
It's
okay.
not
Okay, are there any questions from the audience? Please, Emma.
Just one brief question. When you have infection with chromosomes, you have an immune response. We heard some about the antibodies, which are coming up in previous discussions, but there must be specific T cells coming up as well. CD8 cells with specific receptors for some type of chromosomal antigens, and CD4 cells, of course, I suppose. And I mean, this must, of course, when these cells then meet the antigen, then they will, among other things, produce lots of interferon gamma and stimulate parasite growth, maybe. So I just wonder, have you ever looked for specific cells and how they behave in the course of this infection with chromosomes?
No, actually, we haven't done that. It would be interesting. I mean, now one should do a lot of further studies and look for different things.
Do you know whether or not CD8, specific CD8 cells exist in these rats? Specific for some pattern somewhere. And this is, I think, of some importance also from the vaccine point of view.
Other questions? Okay, if not, we thank first of all Marie devoting time and energy for this nice scrutiny of the thesis, and the committee for spending time here, and the audience too. And then now there is a meeting with the committee to decide this. Thank you all.